So welcome to the next class on classical genetics. So in the class la vande நம்ம என்ன பார்க்க போறோம்னா monohybrid cross, test cross, reciprocal cross, and we'll try to understand the molecular mechanism that involved in determining the plant's height. So let's begin. So first the topic comes monohybrid cross. So in monohybrid cross நம்ம என்ன பார்ப்போம்னா we are just going to breed plants for a single trait. சரி, ஒரு single trait வச்சு நம்ம inbreed பண்ணுவோம் இல்லையா? So monohybrid inheritance is the inheritance of a single character. That means we are just taking care of, or just we are observing, or we are taking a just single character, ஒரே ஒரு character தான், right? And that one character can be any of the character of the plant. For example, வந்து நம்ம pea plant ல 7 characters இருக்கு இல்லையா? Like plant height, pod color, seed color, seed shape இருக்கு இல்லையா? So any one of the character, right? And these characters are found in homologous chromosome, right? They are available in homologous chromosome la irukum. For example, let's take an example for height, plant height. So inga irukku enna irukku, one vandu caps t irukku, that is dominant, or we have a small t which is recessive, right? So idu rendum edula irukku, homologous chromosomes la irukku illaya? That's important to understand. Homologous chromosomes la irukku. So these two are homologous chromosome carries them illaya? So see, there are two alleles rendu da irukku, but the character they are carrying or possessing is single character, homologous. Right? So enna paakano, it's a inheritance of single character, Then what we need to understand, they are in homologous chromosome. Those two alleles, that is for example the plant oda height eduthukodum. Homologous chromosome la irukum, right? Okay, then let's talk about the Punnett square. Okay, so Punnett square was developed by a British scientist, and his name was Reginald Punnett. I hope you know. So Reginald Punnett. So Reginald Punnett was a person who gave the Punnett square. So Punnett square is actually a square diagram that is used to predict the genotype of a particular cross or breeding experiment. நம்ம யூஸ் பண்ணுவோம் subsequent அடுத்த ஸ்லைட் வரும்போது Punnett square யூஸ் பண்ணுவோம். So let's try to have a brief look on Punnett. So Punnett was a British genetist. He was a British genetist, right? Genetist, born in 20th June இதே மாசம், right? 20th June in 1875. He studied in University of Cambridge. He studied. He was the first person to wrote the first book on genetics. He wrote the first book on genetics, importantly. He was the first person to wrote the book on genetics. First person to wrote a book on genetics. That's interesting. Today we have so many books on genetics. He was the first person. Genetics, right? And the name of the book was Mendelism, which was published in 1905. And do you remember that the person's name the person who gave the name coined the term genetics? Yeah, he was William Bateson, right? So he was a collaborator or a friend or a co-worker of William Bateson. He's collaborator of William Bateson. Right, that's interesting about Punnett. Okay, now next go to next topic. Okay, if you want to know more about Punnett, the link I put it here. Okay, you can go and check. You will get the whole biography of original Punnett. Okay, then reciprocal cross. In reciprocal cross, what we do is that suppose we have two individuals, male and a female plant with genotype of tall and for dwarf, that's the capital T and small t, and we interbreed them. If we interbreed them, we get a hybrid plant with a heterozygous genotype. But what if we just change the gender with the genotype of the plant? That means if we cross a dwarf male plant with tall female plant. So in the condition 1, we got a tall plant, right? So what about in condition 2? Here also we are going to get the same, yes, the same heterozygous dominant individual, right? So this kind of cross in which you are changing the gender with the genotype, that we will call as a reciprocal cross. Okay, so if we just self cross this particular individual, right? So if we self cross this individual, what we will get? Let's check it out. Okay, so we are trying to self cross this individual. So we have anther, right? This plant's anther is here, so anther, this is anther, and you know anther is the male part. It's the anther which is the male part of the flower, and then we have a stigma. Here. Okay, stigma here. Okay, and then we have ovules, right? Ovules possess the egg cells, so this will be the female part, right? And then let's try to cross them, right? And see, you know, this type of square, the square shaped structure, that we call Punnett square, right? So this was first designed by Punnett, so we call them Punnett square. Punnett's square. Okay. Right. Why it's a square? Why not a rectangle? Because the number of genotype you are taking from the female, equal number of genotype can be go for the male also. So it's going to be the same number. That's why it's a square always. Right? Okay. Okay. So then from the anther, the two alleles, and from the ovule again two alleles, and it's a random mixing. So let's see if these two alleles gets mixed up. If these two alleles get mixed up, then we get capital T. That means it's a homozygous dominant condition. Okay, so if we get this one with this one, then we get a heterozygous dominant condition, right? And if we get this one with this one, it's a heterozygous again dominant condition. And if these two recessive alleles get mixed up, then we get a homozygous recessive condition. Okay, let's check that how many kinds of genotype we got. Okay, so let's take the capital T. How many individuals? 1. Then capital T small t, that is Heterozygous dominant condition we got 2, then small t and small t. Homozygous recessive condition 1, right? So okay, so what do you think, what will the color of these 3 individuals? Definitely there is always a dominant character here, right? There's always a dominant character here, at least one dominant character. That means what? They are going to be tall. And this one is going to be dwarf. Right? So this will be the genotype. This will be the genotype. And this will be the phenotype. So if we want to make the ratio, then the ratio will be how much? For tall, how many individuals are there? For tall, there are 3 individuals. For dwarf, there is a 1 individual, right? So if you try to make the ratio, then you get 1:2:1. That is tall, homozygous, heterozygous, then again homozygous recessive, right? And since they are all tall, which is 3, and this only one is dwarf, it's 1, so ratio will be 3:1, right? So remember that the phenotype ratio is 3:1 and genotype ratio is 1:2:1. That was very basic actually. Okay. Okay, then next is Mendel's analytical and empirical approach. So interesting fact is that Mendel chose 2 contrasting traits, right? And that was interesting because he noticed that he noticed that for a single character, or for a single phenotype, there exist different traits. There exist different traits. So what I want to say is that if you take tall as a phenotype, these 2 traits exist, right? What are the traits? The tall itself. Let's take height of the plant. Okay, don't say tall. Let's take height of the plant. Okay, let's take it as a height. That will be appropriate. Okay. So let's take height of the plant. So if we consider height as a phenotype, so there exists 2 different traits, that is tall and dwarf. He noticed that, right? And what he noticed, he also noticed that in F1 generation, now the recessive trait disappears. The recessive trait disappears or hidden or masked. But interestingly, in F2 generation, the recessive trait reappears, right? And what ratio? It appears in the ratio of 1/4 of the F2 generation. It appears in the ratio of 1/4 or 25% of the F2 generation, right? That was interesting. Okay, so with that, Mendel was able to understand that in F2 generation, the ratio for dominant and recessive comes in 3:1 ratio for genotype, right? Sorry, for phenotype. Okay, so Mendel noticed that in F2 generation, the phenotype was 3:1, that is 75% and 25%, right? So by this, Mendel concluded that the tall allele and dwarf alleles segregate randomly. He concluded that the tall alleles and dwarf alleles segregate randomly, but not in a particular fashion, but just a random segregation. And I hope you remember that gave the law of segregation randomly, right? So using mathematics, understanding the ratio, so Mendel was the first scientist to do quantitative analysis, right? So he quantitatively checked the how much of the individuals possess the dominant character, how many of the individuals possess the recessive characters. So he was able to bring a pattern, right? So we can consider his search or his study as a quantitative approach, right? So the next thing is that Mendel was able to understand that these characters were carried out by alleles, which he also called as a particulate, or he called them as a discrete particulate unit of heredity. He called them as a discrete particulate. The particles that are carrying the heredity, particulate unit of heredity, and what we call them today. Do you remember what we call this discrete particulate unit of heredity? Today we call them as genes, right? And what they do, what he did, so by his experiment, Mendel was able to understand that traits were carried by the alleles, right? And he tried to understand the relationship, that what kind of allele brings what kind of character. He tried to understand their relationship. Right? And that create the empirical approach. Right, which later became the laws. Okay, we'll see that later videos. Okay, so now next one is the test cross. So in test cross, what we do, so see, test cross, the word itself says is a test of something, right? So actually the condition is that in a test cross you have an individual, right, with unknown genotype, right? You have an individual with unknown genotype, there's an individual with unknown genotype, and how we can find its genotype, right? So we have unknown genotype individual with unknown genotype, and we want to find the genotype of this unknown individual, so unknown genotype individual. So what we do, we cross them with a homozygous recessive individual. We cross them with a homozygous recessive individual, and if we do that, what will happen? Do you remember? Let's check it out. Okay, so let's consider the two conditions if the individual is a plant with heterozygous dominant character. Now heterozygous dominant, you remember, I believe that heterozygous dominant, for example, if we take for tall, it's one capital T and one small t. That means one is dominant, one is recessive, so it's a heterozygous dominant condition, right? Dominant, and the other one is recessive. But still this plant will appear tall, so you don't know that whether this plant is homozygous dominant or heterozygous dominant. You don't know that. Okay, so to find that whether it's a heterozygous dominant or homozygous dominant, what we do, we cross them with recessive homozygous recessive individual. So let's see, so what happens. We already checked that. So if the individual is a homozygous dominant, okay, so if the homozygous dominant, then you see the genotype ratio you get is is 1:1, right? Right, so if you cross these two, you get this one. If you cross these two, you get homozygous dominant, and again if you cross these two, oh, sorry, okay, so if you cross these two, this one and this one, you get a homozygous embryo. If you cross this one and this one, you get heterozygous dominant individual. Then if you cross this one with this one, so you get homozygous recessive character, and if you cross these two, then you get heterozygous dominant. Okay, so if we take 4 plants, then how many plants with genotype heterozygous condition? You see there are 2 plants, and if you see a plant with homozygous recessive condition, again you see 2 plants. That means the ratio is 1:1, right? So genotype ratio is 1:1, right? And for genotype ratio 1:1, the phenotype ratio is also 1:1. That means you get 50% plants tall and 50% plants dwarf, right? So we have 50% plants tall and we have 50% plants dwarf. If such condition comes, that means if the F2 generation is like this, if the F1 generation is like this, then you have to understand that the unknown plant is heterozygous dominant. It's a heterozygous dominant plant, right? Okay, the condition 2, let's see the condition 2. In condition 2, the plant is homozygous dominant. It's unknown, right? It's unknown. We don't know that. Okay, so then let's cross it. If we cross, what we get? We get the heterozygous dominant character. Again, when we cross these two, and we cross this one and again this one, we get heterozygous dominant condition. Okay, and if we cross this one with this one, again you get the heterozygous dominant condition, and if you got this one with this one, again you get heterozygous dominant condition. So this combination is all right. The all individual possess this character. That means all plants are tall, right? So all plants are tall, right? Both possess same, so tall. All plants are tall, right? So if in F1 generation, you get all tall plants, right? If in F1 generation you get all tall plants, F1 all tall, that means the unknown is homozygous. Unknown is homozygous dominant. And if the F1 generation is 50% tall and 50% dwarf, then what's then? Then the unknown is heterozygous dominant. Then the unknown is heterozygous dominant. Clear? I hope so you're clear. Then why Mendel's pea plant are tall and dwarf? That's a good question. I also used to puzzle that why some plants are tall, some plants are dwarf, right? Okay, so before trying to understand, I want to take you back to a chapter called growth and development in plant. And if you remember that chapter, there was a condition, or there was a term came like Bakane disease, right? The Japanese farmers saw some rice seedling which was very tall. Do you remember that why it happened? Do you remember? So let's see that how they look like actually. So see, generally when the rice seedling heights is like this, A B C D. Okay, the moon all blue color bucks analytical plants like this short arco. But some plants like red leaf, yellow leaf kamadi, they are tall. Yeah, why they are tall? So later they found that there is a fungi called Gibberella fujikuroi which infects them, and these fungi produces a kind of growth hormone, a kind of plant hormone which is known as gibberellin, right? So when you spray gibberellin even in a dwarf plant or short plant, okay, they show elongation in their stem length. So if there is a dwarf pea plant and you are spraying gibberellin in dwarf pea plant, you can see that they become tall. Like dwarf pea plant treated with gibberellin, that means gibberellin has to do something with the height of the plant. Okay, in later period when researchers become aware of new molecular techniques, they try to find what will be the reason behind that, right? Okay, so basically let's try to understand a basic concept that we have a DNA, right? And DNA, what it does, it get transcribed. Okay, so this DNA is a part, right? Let's take from here. Okay, it's a part, right? Part of the DNA, part of the chromosome, right? Okay, so it's transcribed, and after transcription they produces RNA, and these mRNA get translated and makes the protein. These proteins are folded to become an active protein, right? These amino acids are folded to become an active protein, and what they do some function in metabolism. For example, we have a substrate. Okay, they catalyze those substrate reaction and makes us to get the product, right? For example, this enzyme comes and they just helps us to get the product. Okay, so in our length condition, you see there is a gene called LE, okay, for length gene. Okay, and this gene produces the mRNA, right, for a particular enzyme, right? And this enzyme, what happens? Okay, so this enzyme, what it does, okay, it converts the precursor of gibberellin into gibberellin. The precursor of gibberellin, GA20, is converted into gibberellin by this enzyme gibberellin 3-beta hydroxylase. Right, and what this gibberellin do? Okay, they promote the elongation of the stem's length, right, and because of which plants becomes taller. Right, so that was a story of plant which possess the allele. That means the plant which have the CAPS T allele. Right, if the plant has a CAPS T allele, they become tall, right, they become tall. So in case, okay, if you want to know more about this thing, this link is given, you can go and check. Okay, let's check it out. That if this is a homozygous dominant, right? So homozygous dominant condition, there's 2 capital T. So what happens? We had 2 alleles, CAPS T and CAPS T. Okay, these 2 alleles are coding for the allele gene. Then the system will produce the 3-beta hydroxylase enzyme, right? And what these enzyme will do, these enzyme will convert or produce gibberellin, and because of which the plant will be taller, right? The plant will be taller. So let's see in heterozygous dominant condition. So what happens in heterozygous dominant condition? So when we have one functional allele, right, so this can code for gibberellin 3-beta hydroxylase, and they cannot make 3-beta hydroxylase. Okay, let's see that what happened. So we have, this is non-functional, this is non-functional, right? So this is non-functional, so this one functional is functional. So what the function will do, it will create gibberellin, and because it creates gibberellin, the plants will show an increase in height, right? So we can get a tall plant. So when it's a heterozygous condition, you get a tall plant because of this reason. Okay, then let's see that it's a homozygous recessive condition, right? If in a homozygous recessive condition, what happens? So see here, both alleles are small t and small t, that means both of this allele is not going to make a functional enzyme. So it's no functional enzyme, here also no functional enzyme. Right, so since there's no functional enzyme, so even though you have the precursor for gibberellin, but since there's no functional enzyme, you are not going to get the gibberellin. Since there's no gibberellin, so plants is not going to increase in their height. So plants is not going to increase in their height, so they are going to stay dwarf, right? They're going to stay dwarf, so they will be dwarf. So when you have the small t allele, okay, there is a loss of the functional enzyme, and since there is a loss of functional enzyme, the plant cannot make gibberellin. So if it cannot make gibberellin, they stay shorter. But what about if you spray some gibberellin exogenously? If you spray some gibberellin exogenously, definitely they will grow and they will be similarly tall. But that is just artificial, not natural. Okay, the next is back cross. Okay, so what happened in back cross? Let's see. So back cross is a cross of F1 hybrid. It's a cross of F1 hybrid with any one of the parental genotype. That means that parental genotype can be a maternal genotype, or can be a paternal genotype, right? Both, it's possible, right? So okay, let's see an example. So we have a plant, right? 2 plants, okay, and we are crossing them. So one is with homozygous dominant trait, other one is homozygous recessive trait, and when you cross them, definitely you are going to get a hybrid heterozygous dominant trait, right? So if this heterozygous dominant trait you cross with the same genotype, parental genotype, for example here we are crossing it with a parental genotype, right? Either the tall genotype or a short genotype. Okay, so that condition we call as a back cross, right? So let's see type of back cross. Back cross are generally two types. One is dominant back cross, the other one is recessive back cross. In dominant back cross, what happens is that if you are, or if the offspring is crossed with an individual having dominant parental genotype, right? If the F1 generation is crossed with a dominant parental genotype, what happened? All the F2 generation will show the dominant character and no recessive individual are obtained in the progeny. So let's check, does it happen? Okay, so we get the F1 generation, right? So this F1 generation individual is crossed with dominant parental phenotype, right? So this is the dominant parent, and this is the recessive parent. Okay, if this F1 individual, is crossed with a dominant parent's phenotype, what do you get? Just think about that. What are the genotype will get? Caps T again, then small caps T small t and caps T small t, right? So F2 generation will be tall, and tall. Genotype is given, right? So what you see here is that the F2 generation possess homozygous dominant, two numbers, and heterozygous dominant, two numbers, right? So what's the ratio? 1:1. What is the phenotype? All tall. So genotype ratio is 2:1. In such condition, genotype ratio is, I will say that this 1:1, and phenotype all tall. Okay, then recessive back cross, when F1 generation is crossed with an individual having recessive parental genotype. Okay, so what happens? Let's see. So we have the F1 generation, right? F1 generation, and this F1 generation is crossed with recessive parental phenotype, right? Recessive parental phenotype could cross now. Just think what you get. You have, finish this. Okay, so what we get 50% individual with dominant character, 50% individual with recessive character. So எப்படி இருக்கும் genotype எப்படி இருக்கும்? Genotype வந்து 1:1 இருக்கும். Genotype எப்படி இருக்கும்? Genotype, if we take capital T small t versus small t no பார்த்தோம்னா is 2 and 2, that is 1:1. Ratio. And if you see phenotype ratio, is also same. Yeah, phenotype ratio pathalume, when you see phenotype ratio tall versus dwarf, pathona there are 2 tall and 2 dwarf, that is 1:1. So both genotype and phenotype are same. That means 50% tall and 50% dwarf, right? Okay, interestingly, The recessive backcross it able to identify the heterozygosity of the hybrid. Therefore, it can be considered as a test cross also. Because of this, you can find that whether the individual in the individual வந்து homozygous heterozygous உங்களுக்கு தெரிய வைக்கும். அதனால வந்து recessive backcross can be also considered as a kind of test cross, right? So with this I am finishing this video class. We'll meet in next video class. I hope you understand. If you have doubt, feel free to mail me, right? Have a great time. Okay, with this I am finishing this video lesson. We will meet in next video lesson. In case if you have doubt edha varudhu puriyala appdi solumbodhu, feel free, mail me. Mail ID kuduthirukku, mail pannunga. Seriya? Bye.